was really a fantastic introduction. Thank you very much, and thank you all for being here. So, I am an engineer, and I was always more interested, however, in discovering things rather than simply building them. So, when I was studying at university, we had this joke with my parents. They were always telling me, if you want money, then you go to industry. And if you want glory, go to academia. So, well, I'm happy to tell them that they were wrong. Because while it is true that I don't have much money, I also don't have achieved much glory so far. So, since it became clear some time ago that both time and glory were out of the question, I decided to focus myself on something much simpler. I decided to go and seek the truth. So I thought, well, it's easy, no? Well, it turns out it's not so easy. I'm not going to get too philosophical, what is truth and how you define it, but fortunately in medical sciences and in a lot of sciences, we have a tool that can help us, and this tool is statistics. So we don't have to care about philosophy when we have statistics. So imagine that I find a family of stray cats, like this, and I want to adopt them. But actually they're stray. I don't know if they're nice domestic cats or if they're wild cats. And wild cats cannot be tamed, so I definitely don't want wild cats. So I weigh them on a scale. So I know that cats, domestic cats weigh 4 to 5 kilograms, and wild cats weigh 5 to 8 kilos. So I measure the weight. I have these cats, I measure their weight. So I can measure the weight and say maybe some are 6, some are 5.5, and I can say I think or I don't think that these points or these weights come from this distribution. So I know that the domestic cat range, and I measure my cats, and I can say, okay, they seem to fit in the domestic cat category or in the wild cat. But actually I can do something better. I can have a test that tells me the probability that I have when I put this data in my algorithm, the probability that I have that I'm wrong. So I can assume that they are wild cats, and I can measure the probability with which I am wrong. So of course, in this case, you know, the farther the cat's weights are from the mean, then the probability that I'm mistaken in considering them as wild cats goes down. No? So if they're here, then there is a high probability that I'm mistaken, that these are wild cats and they're domestic cats, but if they're here, they progressively become lower. So at some point, the scientific community decided that if this probability was like a ridiculously low number, then the statement, these animals are wild cats, is not just probable, it is true. So we just say, okay, if this number is really, really small, then let's not lose ourselves in technicalities. Let's say it's true if this number is very small. So this also applies, of course, not just to cats, but also to a drug or to an imaging method. If the probability that this drug doesn't work is very, very low, then the drug works. If the probability that my method also doesn't show what I think it shows is very, very low, then it must be true. So truth in our case is not absolute. So it just means that this thing has a very low probability of being false. So what is this ridiculously low value that tells us that something must be true beyond reasonable doubt? So this ridiculously low value that we all somehow agreed upon is 5. Actually 5%. So it means 1 in 20. Do you know what 1 in 20 means? Well, this is 1 in 20. And I have it here. So let's chance that one specific number on this die comes out in a row is exactly 5%. So is it a ridiculously low number? Well, let's try. You tell me. Let's make a game. So I ask one of you, one volunteer, rolls this die. You choose a number. One volunteer there. You choose a number. If the number comes out, so you have 5% probability, you give me 2 francs. If it doesn't, I give you 2 francs.
I say 20.
You say 20, okay. 14. No, it's good. You get 2 francs. Congratulations.
Oh, wow.
Another volunteer challenges me for 2 francs. Yeah, Christoph. It's easy money, no? You only have 5% probability of losing. 5? Oh, it's a 2. I also lose 2 francs to Christoph. Okay, so far they have won, no? They had 5% probability of losing and they have won. But they were only betting 2 francs. If we repeat now this, if the number doesn't come out, I give you 2 francs. If the number comes out, you give me 500 francs. Do we have some volunteers for 500 francs? You should say no, otherwise this doesn't prove my point. So you see, this ridiculously low number is strange because it's pervasive everywhere. We call it significant, but we actually mean that it's true. And if we want to publish something, then we always aim for lowering our error below 5%. But actually, if it's low or not, it depends on the stakes. So imagine that now we've played with some money for fun. I lost 4 francs, and I will remember this. But imagine that I have a treatment for gray hair. I mean, most people don't want gray hair. And I do a study, and I tested it, and I tell you, look, this is a treatment, it works, and the probability that it kills you is less than 5%. So it's basically nothing, no? In a paper, this would mean that it doesn't kill you. Actually, you have lower than 5% probability. I don't think that you would be happy to have this treatment for gray hair. I would personally keep my gray hair or my losing hair here. So, an arbitrary value, I don't think it makes sense. I think you can all agree that it makes sense. And we should, first of all, try to adapt this threshold, but this is the reality. How ridiculous it is also shown in this very nice comic. The aliens come here, super advanced, and the men ask, what is truth? And the aliens say, probability below 0.03. Notice that they only have 3 fingers in their hands, so maybe this gives a hint why people chose 5 instead of 3. But we decided, so originally this was never meant to be, of course. A statistician came up with this number, but it was kind of an estimation. Let's say, okay, let's take 5 so that it's a reasonable number. Then we have to look more into it. But now with a scientific community moving on, this has become kind of engraved in stone, and we always look for 0.05. But why is this bad? So the problem is that just like rolling a die, any random experiment will turn out true 1 time out of 20. And if I repeat the same experiment 20 times, even if it doesn't work, it will come out true. And if I repeat 20 different experiments, statistically one of them will achieve this 0.05. So if we want to publish nowadays, or well, up to some time ago, but mostly still, we need to achieve this 5% threshold. But this caused the problem that we have the incentive to look for this 5% threshold. And if we don't publish all the 19 studies that came out above this 5% threshold, and we only publish the one that are below, then we don't know if what we publish is actually true or false, because it can just be a fluctuation. And this already in 2005, but actually even before, some scientists made even the bold statement that most published research findings are false, because we are selecting for this 5% threshold, but we can achieve, just like rolling our die, we can achieve this just by random chance. So this to say that my quest for the truth, after losing money and fame, I also had to drop my quest for truth. So in recent years, I stopped trying to find things that are true, and I moved to things that are reproducible instead. And this has become my mantra or my guiding light in the last few years, because this means that repeating the same experiment by another person, hopefully they will get the same results. I don't know if they're true, but at least if I find something and someone else finds the same thing, at least there's some indication that they can do that. So there are many aspects related to reproducibility. Of course, this was statistics, but statistics is just a small part. People cannot reproduce my experiments if they don't know how I did my experiment. And so, since we are in the method development business, what we want to do is to make methods that are reproducible. And this is especially important if we're talking about experiments that involve a small number of subjects. So, you can imagine, I will not go into details, if I run a study on 1,000 people, it will have a different value than if I run a study on 10 people. But this brings us to the scientific topic that I've dealt with for the last 10 years, more or less. So now we'll talk about muscle imaging, which is actually what you mostly came here for. Statistics is my passion, but muscle imaging is actually the core. And why is it important? So muscle imaging, we do it because we're mostly focusing on neuromuscular diseases or any kind of disease that has some muscle involvement. These can be direct diseases that affect the muscles, like dystrophies. They're usually genetic diseases. But they can also be neural disease, either in the central nervous system or peripheral nervous system that has some effect on the muscle because of atrophy or wasting. And if we're talking about neuromuscular diseases, now this low number problem comes into focus, because if we compare it to something like multiple sclerosis or Parkinson's disease, in general, neuromuscular diseases have quite a high prevalence. So there are quite a few people affected by these diseases, but each single one, if we want to study them one by one, then we see that the numbers are actually very small. So in our hospital, if we are focusing on a particular dystrophy, we might have maybe 10, 20 possible patients, and if we want to run a study, of course, not everybody is able or willing to participate in this study, so we are looking at small numbers. So we need to have methods, and this is where we come in, so me and my team, that other people can use, other people can apply, so that we can really reproduce our studies and our findings. In other countries, this is not the case, or some countries, I have some collaborations in China, and I was in China some time ago, and they have hospitals with hundreds of patients with a specific dystrophy. And they are very interested, of course, in doing studies with our methods, and they can run a single center study. But in our case, it's very important that we can run a study in a multicentric environment, and so that we can share our methods. So sharing our methods is really become our modus operandi. What are we looking for, however? So our methods are focused on this degeneration pattern of the muscle that is usually ending with fat replacement. So we start with a healthy muscle tissue that has fibers, and they are inserted in an extracellular matrix, which is like a scaffold where these fibers act upon. And if something happens, either through direct damage of the muscle or through just muscle wasting, you see in the diseased muscle that you have fat replacement. So, of course, assessing how much fat we have in the muscle is a very important feature, and also assessing this kind of damage before fat replacement occurs is also important. And this is the spectrum of methods that we have available. In general, we see images like this. So these are the images that neurologists and neuroradiologists look at. These are qualitative images, and here you can qualitatively evaluate. In this case, the hamstrings are basically completely replaced by fat. So this is a typical presentation. And in other cases, this is a stir image. This shows the amount of inflammation that there is in a tissue. And so we see that some areas of the muscles can be highlighted with respect to the others. But this is the classical way. Of course, these methods work. So we have been focusing more on quantitative imaging and on dynamic imaging. And as I said in the beginning, this cannot be done by us alone. And I want to underline already that we are part of a big network of people and these two very important people already, I want to make a shout out to them, are Anna Picchiecchio from Pavia and Giorgio Tasca from Rome, now in Newcastle. And the funny story here is that this started already maybe 7-8 years ago. I was invited to go to a meeting called it was a small meeting of the people doing research on muscle. I was an outsider because, as you heard, I first came to this world from a heart perspective. I was working on heart, and somehow heart and skeletal muscle are quite different in terms of communities. So I was invited to go to a meeting of muscle people. And I was presenting one of the methods that I'm going to show you in a moment, and Giorgio was there. And it was this dynamic imaging method. We've been working on dynamic imaging for many years. And Giorgio saw the method and told me, yeah, we need to apply this in patients. And I told him, look, Giorgio, no, it's nice, but it's not ready. It's really the start. We still have to validate. No, but it's really interesting. It's really interesting. And you have to know something, and this I'm telling a secret, but we're not too many people. Sometimes medical doctors are very enthusiastic when they come to you with a research proposal, but they don't always follow up on it. So initially, I always tell them, yeah, okay, let's talk about it in a week or two. And usually they don't come back. He did. And he was calling me on my phone and said, yeah, look, I think I found a way because... And I was telling him, maybe it's still not ready. And he insisted, and he kept calling me every 2 weeks until eventually he said, look, I found Anna in Pavia. She wants to do the study. We have the patient. She has the machine. And this was actually the beginning of a very beautiful and fruitful collaboration. So if I'm here today, I definitely owe it to these people that insisted so much that I tried a method that I thought it was not ready. But it took years before we actually got results there. And they worked a lot, and they worked very hard. So this is the network. And of course, we have collaborations here in Basel. We have collaborations with the Department of Sports. And we have method exchange with Leiden. And of course, our open methods are used everywhere. One of these methods is a quantitative assessment of water T2. So water T2 is one of the fundamental properties physical properties of tissues, or actually any object. And the T2 is correlated with edema. So you can imagine that when you measure T2, you're basically measuring the amount of edema. In muscle imaging, this is very important because we've seen that it's one of the stages where we have inflammation, but it's actually quite complicated because of this fat replacement process. So I will not go too much into detail, but we developed this method that is based on some theory that also Matthias helped develop, Matthias Weigel, who is sitting here. We did a very nice open source version, and this was still not very common to have open source implementations of these algorithms. Everyone had their own, more or less, but there were also a lot of people doing research who did not have the knowledge to build the methods themselves. And so we published this already some years ago, and the images that we obtained were quite interesting. So you see here, for example, in these muscles, the edema was highlighted, these green parts with respect to the blue, whereas this is a volunteer that has no edema. So it worked, and we developed it, and actually this had quite nice impact, not just from our collaborators. This is just a small selection of papers that then took these methods. Some of them I don't even know them. Some are collaborators, some are I don't know these people very well, for example. It's very nice that they just saw what we did and they took it and used it for their own research and found something quite interesting and important with it. And currently, this is also, this will become part, or is becoming part of a project that Claudia Weidensteiner here is leading, and Jessica is working on, for the development of fully reproducible pipeline for muscle evaluation. So we are developing together these kind of tools. On the other hand, also also another tool for the community that I've been working a lot on is Daphne. It's a segmentation, and muscle segmentation is an important problem because we want to extract information about each muscle. And segmentation means, you see, highlighting automatically which area of the image corresponds to which muscle. So we can then say the gastrocnemius has this much fat infiltration, or the soleus has this fat infiltration, and this is very important. And we developed this already a few years ago. This is growing as a project. It's moving beyond muscle. Now we have Valentina, who is a PhD student from Italy, who is here working on this together with me. And again, this was a big collaborative project, and we released it to everybody. And now the impact is that we have 176 users, I checked yesterday, and our segmentation models were downloaded almost 4,000 times. So 4,000 segmentation, 4,000 people or 4,000 accesses were done. And as you see, there are a bunch of papers that again are using our methods to achieve their own research. So replicating not just what we did, but also using them in their production. But now we ask ourselves, we've seen images, we've seen these methods are used for numbers, but is an image enough? So when you think of a muscle, you don't think about some static thing. So I ask you, is there some cyclist in the group? I know Philip is quite used to cycling. Is this a good bicycle? It's a picture of a bicycle. It looks okay. I mean, this guy is jumping. But what happens now? So this bike apparently had some problems, but we could definitely not see the problems in a static pictures. We could only see the problems of this bicycle when it actually started performing the function it was made for. So I went back, and this is the method that I was talking to you about, I was coming from my heart background, and in the heart, we don't stop at the look of the heart. We also want to see how it works. So for heart imaging in MRI, we have a lot of applications where we see the image of the beating heart. Unfortunately, MRI is not fast enough usually to capture the cycle of the heart in real time, and it wouldn't be fast enough to capture a contraction cycle of a muscle in real time. But we have some tricks that we can use, and these tricks are the synchronization of the acquisition with the ECG, with the electrocardiogram. So the heart has this internal pacemaker that makes it contract periodically every second, more or less. And we can record it, and we know that the contraction will be repeatable and periodic, and that's how we can do an acquisition. But I thought we don't use this for the muscles because usually skeletal muscles don't contract periodically. But what if we could make them contract periodically? And this was born, the idea of using an electrical stimulator to actually stimulate the muscle and use the same signal that we use to stimulate the muscle also to trigger the acquisition. So instead of having our internal electrical signal, now we have an external electrical signal that is making the muscle behave a bit like the heart and contracting periodically. And this was already a few years ago, and of course the biggest shout out is to Xenia, my wife. We've worked on this technique for years together. And but at the beginning, also Michele and Michele Panzini and Tilman Schubert were pivotal in the delineation of this. And also later we had some collaborations with Anna Hirschman, who was a radiologist here in the hospital. So this was how it was born, and this is what it looked like. So we were applying these electrodes to the leg initially, and we see that we can have this very nice periodic contraction. And thanks to this, we could measure the movement of the muscle inside the scanner. So this is a depiction of how every single element of the muscle moves during the contraction. And we can have a very good temporal resolution because this contraction only lasts a few milliseconds, and we can still capture how every single element of the muscle moves. So this is what it looks like in a static frame. This is also the image that was in my first slide. And to go one step further from this velocity, what we could do, we could calculate the deformation of the muscle. So we want to see how the muscle actually contracts and how much it deforms, and if there are parts that deform and parts that don't deform, for example. And we can extract some interesting values, like, for example, the strain, or how fast you reach the contraction peak, or how fast you go back to its quiet state. And this is an image of the strain, so really the deformation. This is what it looks like in the thigh. So this is the vastus lateralis, one of the muscles of the thigh, and you see that the yellow means that there is a strong deformation. So you clearly see where the contraction happens, and we can quantify this. So we could quantify, and this was the first study that we did. We tried it on healthy volunteers who were young and older, and we wanted to see how these quantitative parameters related to the deformation changed in terms of due to age. So this was not really new. Some people had measured it before, but we wanted to see if we could see it with our method. And we could clearly see that for the same stimulation intensity, the senior volunteers had a much lower strain values than the young volunteers. So this motivated us to go on and try to apply it in patients. And our first study was on facioscapulohumeral dystrophy. Again, this was the study that Giorgio and Anna pushed a lot. And we studied this longitudinally, and we didn't see such a clear pattern because at the time we didn't really have a clear hypothesis of what the behavior could look like, but at least we could show that imaging this kind of contraction was feasible in this patient population. So this is a dystrophy, so it's a genetic condition that affects the muscle. And we saw that there were, for example, some patients in which the strain values decayed significantly over time, and other cases where these strain values didn't decay. And this was somehow correlated with also some clinical findings. But then we did a more accurate investigation, and this is newer results on a small cohort, but at least now we had more or less a clear hypothesis. So we followed up on what we had did on facioscapulohumeral dystrophy. This was a more diverse patient cohort, but what we saw is that there was a clear trend that in volunteers, this on the right side, this is moving on too fast. The strain values were elevated in volunteers or lowered in patients, and going in the same directions, especially in the soleus. We saw that the build-up rate, so the speed at which the muscle contraction reached this plateau, was also faster in volunteers with respect to patients. So in patients, even with the same force output, we had lower deformation and a slower deformation. The release rate didn't have, I'm not sure why this is moving by itself, but the release rate didn't show similar trends, but well, this was okay. I'm actually personally quite a big fan of the release rate as a potential biomarker, meaning the time it takes for the muscle to go back to its quiet state after contraction, but it turned out not to be that significant in this study. It's okay. At least strain and build-up rate seem to be very, very promising. And not just electrical stimulation, we applied similar techniques also to voluntary contraction, and this is also a study that was done by Claudia, the fish study. So you see, Claudia developed this nice program where we applied this in kids, and the kids had to push a pedal and make these fish eat the bubbles so that they could have a nice repeated voluntary contraction. And what we found there is that we applied this on cerebral palsy, so children that were affected by cerebral palsy. And we also compared it with neuromuscular stimulation. And we saw that with neuromuscular stimulation, we could have a much more repeatable contraction than with voluntary. In healthy volunteers, we could also measure a good contraction with a voluntary motion, but for a more complex situation like in cerebral palsy, neuromuscular stimulation seems to be beneficial there. And of course, this is the study that we have done, but to develop this, we had to develop our own hardware. So I told you that here the patient had to push on a pedal, and the pedal was this. This is something that some project that we did a few years ago. It was an MR-compatible sensor that we also built out of off-the-shelf component. This costs maybe 30, 40 euros to build, or francs, money, dollars. And people were quite interested in it. And this worked very well. We did all our experiments. We built another one for Pavia, so they have really the same setup. And people can just go to the website, download the information, download the schematics, and build exactly the same thing. And what is happening now, we are extending these to the grip, because our next project will be focused not just on the legs, but also on the arms. And here we have Sabine Reuber, who is doing her PhD, and she already started in April, and already she was lightning fast together with Marta in building a very nice 3D printed grip sensor and we've been finishing testing it in these last few days. So again, this is another open source application derived from this, and that we are going to use ourselves and let people build. And of course, not just hardware, we are also bringing our openness and our reproducibility in our sequence development. This open source pipeline that I talked to you about contains also some basic method development for MRI, and this is, again, Claudia and Jesse are working on an open implementation of a rather standard sequence for edema imaging, and Marta Magioni, she's a postdoc, and she's working on open implementation of the dynamic imaging sequence. So far we have an open source sensor, but the method that we were using to measure actually the velocities of the legs or of the arms was still proprietary by Siemens, and we don't know if this could be ported to other scanner vendors. But thanks to this project that is being developed now, we will be able to apply this method on all scanner vendors. And of course, a community is important. I think I convinced you of this. I'm very lucky that I'm part of the ORMIR community, and Serena is here, and she started this a few years ago, and I joined. Here we are really trying to bring the standardization and the collaborative spirit in a bigger context. This is not just muscle imaging, this is open and reproducible musculoskeletal imaging community. And one of the big projects that we are working on is to have a standard data format so that we can really have a completely modular pipeline so that people can use various tools, they can exchange the tools, and they can basically run the same studies in different settings and go back to our initial idea of reproducible science, because not just I can do my experiment, but anybody else, even without knowing me, can do it. So to conclude, I think more or less this is the time. I put a question mark next to truth. Do we really need truth? This is a philosophical question. I think that philosophy is not, unfortunately, my specialty. But I think that in my path, I found something that is more important than truth, and which is doing collaborative science, doing rigorous science. We are trying to help the community, and we are trying to advance the knowledge one small step at a time without shortcuts, and trying to just achieve that holy grail of 0.05 or 5% uncertainty, but we are really trying to do things in a way that people can trust upon. And I would like to end by acknowledging some people. So these are all the collaborators. I mentioned them throughout the presentation. I really appreciate all the work that we have done together. I have to acknowledge Klaus and Oli, Klaus Scheffler and Oliver Bieri, without whom I would definitely not be here in Basel. And I have to acknowledge Xenia, because with her I not only she supports and bears me every day, but she really believed in this muscle imaging project since the beginning, and she developed a huge amount of it. And I want to finish this with a dedication. I want to dedicate kind of my career so far and this achievement. In my master thesis, this is in Italian, 2005 in engineering. This dedication was to my teachers, because they trusted me like I trusted them. And since now I'm kind of going on the opposite side of the desk and being a teacher, I want to dedicate it to the students, and may they find teachers that they can trust, and also by whom they are trusted. So I really hope that I hope this to all the students and to all the scholars that are seeking science. And I would like to thank you all for being here and for listening to me. Thank you.